Time Course Inspector is an open source app for analysis, mining, and clustering of time series data. We wrote it in RShiny, and the app can run locally in RStudio environment or can be deployed as a web application. The app accepts CSV files in a long or wide format. Here I will demonstrate it using a biological dataset from single-cell fluorescent time-lapse microscopy. We have around 200 time series from 3 treatments. Each time series corresponds to protein activity measured over a period of 4 hours in single cells. After uploading the file, we can plot the average protein activity over time from the entire dataset. In order to plot the averages per treatment, we use the grouping column present in the CSV file. The 3 groups correspond to 3 concentrations of the treatment. There is an option to calculate statistics of track lengths or measurements. We see a pulsatile behavior of protein activity. This is because cells were stimulated with 3-minute pulses of a growth factor, and the stimulation was repeated every 20 minutes. There are 2 interesting biological phenomena. First, the responses to stimulation with low and high concentrations are pulsatile but shifted in phase. Second, the middle concentration seems to induce a flat line. We will investigate these 2 observations using our app. In the next tab, we plot individual time series. They look a bit strange because we need to make sure that we have a unique identifier of the time series. Such a unique ID was not present in the source dataset, but we can create it in the user interface using an additional grouping column. We switch to the interactive mode to inspect individual time series. This is handy to find out identifiers of outlier time series. Once we store them in a separate CSV file, we can upload the file such that outliers are removed from further analysis. The file should only contain a single column with time series IDs. We slightly trim our time series and we move to inspect frequency components of the time series. The power spectrum density is calculated for every time series and then averaged per group. We need to specify time units between 2 consecutive data points. Here we set it to 2 minutes because that was the sampling rate during the experiment. Again, we switch to the interactive mode and we see that in every group, time series have a peak at around 24 minutes. This corresponds to the 3-minute stimulation with a 20-minute pause. What is interesting is that the middle concentration also has such a peak, even though the population mean is flat and individual time series are very noisy. In other tabs of the app, we can calculate area under the curve and plot it as a box plot per group with additional overlays such as a violin plot or a dot plot. Moving on to the next tab, we can plot measurements at selected time points. Here I selected 2 time points, the baseline and the peak response after the first stimulation pulse. In the Correlations tab, we can visualize the correlation between 2 time points. Here I'm choosing a time point at the baseline and the first peak. On the y-axis, instead of showing the magnitude, we can also plot the amplitude relative to the time point chosen on the x-axis. As you can see, the correlation between the first peak magnitude and the baseline is around 0. This means that before clustering, we can safely normalize our time series to their own baseline. This should help with clustering, and it's quite a common step in this kind of analysis. Finally, we move to Clustering tab. Here we see a warning about missing data points. It is because the original dataset in the long format had some time points missing entirely. Here, data is displayed as a heat map where each row corresponds to a single time series. Missing rows generates NAs in such a plot, hence the warning. The UI allows to set the shade of gray for missing data to better highlight them. We can see that we have quite a few of such data points. We can interpolate them, but we need to specify the interval between data points, and here we set it to 2. We further trim the dataset to focus on the pulsatile features that we will use for clustering. And as mentioned before, we normalize each time series to its own mean from a selected interval on the baseline. The app offers many linkage and distance methods for hierarchical clustering, and you can quickly view the results of clustering with different methods. We can also cut the dendrogram at a chosen level to better visualize clusters. Here I choose 4. We can inspect our clusters by looking at cluster averages and plots of individual time series in each cluster. Here you can see that by cutting the dendrogram into 4 branches, we have managed to identify 4 distinct behaviors. All clusters have pulsatile dynamics, but clusters 1 and 3 are shifted in phase with respect to clusters 2 and 4. We can also confirm the pulsatile component in the power spectrum. Finally, we arrive at a very interesting representation of our clustering. In this stacked bar plot, we can view the percentage of time series from different clusters to experimental treatments. We have clusters 2 and 4 that have the same phase and make up the majority of the 2.5 treatment. Similarly, clusters 1 and 3 comprise 250 treatment. The intermediate 25 treatment consists of a mixture of all clusters with both phases. In the Cluster Validation tab, we can estimate the optimal number of clusters using 2 very common measures: the average silhouette width and the within cluster variance. Both are calculated for a range of cluster numbers. The former should be maximized and the latter should be small. Typically for the within cluster variance, we are looking at the elbow in the plot, where further increase in the number of clusters does not lower the measure significantly more. We see that 4 clusters that we chose before is quite a reasonable estimate here. Another tab in this module allows to validate quality of clustering for a given number of clusters. We can inspect the dendrogram, which is the same as the one displayed on the left-hand side of the heat map. We can also inspect the silhouette plot, which shows the average distance of data points to other points in the assigned cluster. Time series with negative silhouette width are those that are closer to points in other clusters than to points in its own cluster. We don't have too many of such time series here. Finally, we can see the first 2 components of the principal component analysis. Each dot corresponds to the entire time series, and the points are colored according to assigned clusters. What we are aiming for here is the least overlap between clusters. And as we can see, the overlap is quite small. With this analysis, we have discovered that the pulsatile treatment of cells with an increasing dosage of a growth factor leads to a phase shift. Single cells respond in unison to pulses at a low and high concentration. At high concentration, the responses are in antiphase with respect to the stimulus, and at the intermediate concentration, we observe a mixture of in and antiphase responses. We have discovered that using clustering of time series. This is an important piece of the puzzle that allowed us to define and select mechanistic models of this process.